So if we start out with a mom and a dad, and a few kids. See, we've got a couple filled in symbols. Like that. Okay. So pedigree analysis, the first thing I would do is ask, is this skipping a generation or not?
Okay.
So not skipping a generation is present in successive generations. That does not mean that it can't be recessive. It's just the most obvious starting place.
Okay.
Dominant traits will always show up in every generation.
Okay.
Recessive traits can occasionally show up in every generation.
Okay.
So it's a better bet if you see it in every generation that it's dominant. So that's where we start.
Okay.
So we're going to, step one, assume in this case, our hypothesis now going into the analysis of the pedigree is we're going to assume that this is dominant.
Okay.
And we're going to keep assuming that until we find some piece of information that tells us it's not dominant.
Okay.
And if we never run into anything that says it's not dominant, then the conclusion will be that it could be dominant.
Okay.
So, step two. This is always the hardest part. So when explaining how your brain works, like, I know how I interpret this pedigree, but how do I explain the order of events in my brain that go through analyzing a pedigree?
Right.
So I would say the first thing that pops into my mind is this unaffected offspring is interesting to me.
Okay.
Because it doesn't tell me anything to know that they're affected. So we already got the information from the fact that there's an affected individual here and two in the next generation. That's
Right.
what led us to this first point that it's probably dominant. But none of this information between those three individuals tells us anything about their genotypes.
Okay.
Because if they're dominant, as we went through in the review session, there are two possibilities. They're either heterozygous, they've got one copy of the mutation, or they're homozygous. So that's the first question that I have in my mind now, is when I see this parent pass it to some of their kids, does that help me figure out which of those two possible genotypes the mom has?
Okay.
The thing that's, and it's like, again, the thing that's particularly interesting to me is that there's an unaffected kid. So unaffected individuals are always particularly useful because we know, regardless of whether or not you're looking at a dominant trait or a recessive trait, if it's autosomal, we know the genotype of unaffected individuals. What genotype do they have to have if they don't exhibit the trait? Remember, our assumption is that it's dominant because it shows up in every generation.
Would it have to be plus?
Yeah, absolutely.
Okay.
This individual has to be plus if we're looking at an autosomal dominant or a dominant inheritance pattern,
Okay.
which is our hypothesis. So we're going to say that individual is plus over plus. Okay. Now the kid was born at conceived at fertilization, right? So dad's sperm and mom's ovum had to fuse, and each of them was carrying either a plus or a minus. So to get a plus over plus kid, we know that each of the parents have to at least have one plus
Okay.
to contribute. So at this point, we know that dad is plus over who knows what yet. We don't know what the other allele is. We know he's got to have at least one plus because he's got an unaffected kid.
Mm-hmm.
Same is true of mom. has to have a plus. So you combine those two pluses together to make that genotype of offspring.
Okay.
So if mom has a plus to contribute, now we know which of those two genotypes she has to be.
Okay.
Sometimes the eraser doesn't work for whatever reason. Okay. Now the rest of this, my brain was just working for a second, you could probably tell. I don't think we can get the genotypes of the two kids in this particular scenario. There's probably not enough information.
Okay.
But so far everything is consistent with dominance.
Mm-hmm.
So if dad is plus over, oh, we do know his genotype, if this is dominant.
He would be plus over plus.
Yeah, it can't be a question mark. He
Right.
would show the phenotype if he was heterozygous or homozygous.
Mm-hmm.
So dad has to be plus over plus, mom has to be a carrier to produce an unaffected offspring.
Okay.
Right. So we don't know, we can't figure out, here we can figure out the genotypes of the kids then.
So they're both plus over minus.
Yeah.
Okay.
Have to, right, they're affected. So they had to get a minus from mom, and they can only get a plus from dad.
Okay.
Yep. Should we do another example?
Yes, please.
All right. Let me see, I just have to come up with a different example. So that was an affected mom and two affected kids. Let's see. What about...
Everyone thinks that, like, no, I'm not interested. We're definitely not dating.
Come in.
Definitely not dating.
I'll just say-
Anymore.
You can come in. As long as you want to talk about genetics.
Maybe.
So we're just running through examples of pedigree analysis.
Oh, perfect.
Yep. And I'm just trying to invent a pedigree. So bear with my brain for a minute. Okay, that should work. So we've got 4 options, well, at least of inheritance patterns we've talked about in class. That's okay. It's just my family, it's not important.
Oh, it is a
I'm kidding, don't worry about it. That always happens to the little ones. So is this autosomal dominant, autosomal recessive, X-linked dominant, or X-linked recessive? Let me grab a couple other chairs for that. Oh, that's right. Oh, ponder that first. Where's he going?
He's getting chairs.
Oh. Oh, what
It's
is that?
okay. I was just putting my bag down.
It's just sit down. I don't believe in chairs. They don't exist. Do I still have this zap? I love it. Concept?
Yes, right. Yeah, I would have told you. Let's see, I've got one little chair with no back. I'll be right back.
It's yours. It's all you.
Sure.
I already told you I don't believe in chairs.
Okay. He's in you guys.
Alright, people are going to have to sit on each other's laps. Thank you.
I'm just kidding.
Okay. Right.
Oh, yeah.
Would you mind poking the power button on the lower right-hand corner there? Voila. Okay. Just for ease of operation. So we have to figure out, is this autosomal dominant, autosomal recessive, X-linked dominant, or X-linked recessive, or maybe two or more of those? So can we eliminate all three possibilities and arrive at just one answer, or is this ambiguous?
What's the difference between the autosomal and then the X? Like if they're both dominant?
Right, so X dominant, so what's the inheritance pattern difference between X dominant and autosomal dominant? Right, it's one of those things we talked about in the review session. It's autosomal dominant, an affected dad, there's that reciprocal cross idea.
Okay.
And so we're only looking at one cross direction here. We've got an affected dad and an unaffected mom.
Okay.
So we're not going to get enough information out of this pedigree to distinguish whether it's X-linked or autosomal, unless we see the pattern that breaks the rule for X-linked traits. So first, what was the first thing that we did in the last example? Step one, what was the first decision we made?
We saw if it skipped a generation or not.
Okay, so in this case, this trait does not skip a generation. So the assumption then we make is it dominant or recessive?
Then it's dominant.
Right, so our hypothesis, we don't know yet, because sometimes recessive traits don't skip generations.
Okay.
They're weird cases, and I think one of those actually came up in class today. So when you see them not skip, it's more likely that they're dominant than recessive. So we're going to assume this is a dominant trait. So if this was X-linked and dominant, then what would be true? So what would dad's genotype be if this is X-linked and dominant? He's got an X chromosome, he's got a Y chromosome. He shows the trait. Symbols filled in. He's got the disease, which means his X chromosome is plus or minus. Minus. Right? He's got the mutation that causes this phenotype, whatever it is. Okay, so what has to be true of affected males with X-linked dominant traits? Who gets his X chromosome? How many of his daughters? All of his daughters. So that would mean that every circle down here would be X minus over whatever came from the mom, which we haven't figured out yet. But that's just extra work we don't have to do, because what's the problem with this so far?
One of the daughters is affected.
Right, so daughter 3 doesn't have the trait. So that immediately tells us this is not acting as an X-linked dominant trait. If it was, an affected dad would pass his mutant X chromosome to all of his daughters. So, 3. New assumption, or new hypothesis. We've tested X dominant, but that didn't hold up. We've got a dad that doesn't pass the trait to a daughter. So we've ruled out one of the 4 possible inheritance patterns. 3 left to go. So if it's not X-linked dominant, then 3 is we're back to we're still at dominant because we see the trait in every generation. So it's not X dominant. Let's ask if it's autosomal dominant. So can we come up with genotypes for all of these individuals that are consistent with that sort of inheritance pattern. Okay. So after we figured out, in the first example we did, we saw the trait in every generation, so we hypothesized that it was dominant inheritance. And then, what was I most interested in terms of figuring out the genotypes of the individuals?
The one that wasn't affected.
Right. So I focused first on the fact that there are individuals in this second generation that are not affected by the trait. They're the ones that we can usually first figure out the genotype. If you assume that this is, which is not the case here, I'm just saying this for the record, if this was a recessive trait, then the genotype of any unaffected individual would be plus over plus. It's recessive. Or it would be plus over minus. In this case, we've assumed that this is dominant. So if this is a dominant trait, we absolutely guarantee you know what the genotypes of unaffected individuals are. So everyone's nodding, so tell me what the genotype is. If it's dominant, they have to be plus over plus. Because if this is a dominant mutation, then even 1 minus would give you the trait, and they don't have it.
Would the unaffected son have a X plus and a Y genotype?
So we've already ruled out X linkage. So we don't have to think about, so in other words, yes. This son would be normal X chromosome, normal Y chromosome.
Okay.
He's an XY genotype, but we've already ruled out X-linked dominance as an inheritance pattern. Because we saw this affected male not passing the trait to his daughter. Right? So we know it's not on the X chromosome. And that brought us to the hypothesis that maybe this is autosomal dominant. Now the problem is for the rest of the kids, for the affected kids, could be heterozygotes, could be homozygous minus. Either of those genotypes would give them the affected phenotype. So now we have to work on, can we figure out the genotypes of the parents? So first we started with the unaffected kids. We know their genotypes. We're going to try to figure out the genotypes of the parents. If we can do that, then we should be able to figure out the genotypes of the affected kids.
Should the mom be plus?
Right. If this is dominant, then any unaffected individual we know is homozygous wild type. Okay, so where did the mutation come from? From the kids' perspective, who gave them their mutation? Which parent?
I would say the father.
Yeah, it has to be the father.
Yeah.
Okay, so the only remaining question, well, there are two remaining questions. What's his genotype is the first remaining question. It has to have at least 1 minus. Does he have 2 minuses?
Or else the daughter would have.
Right, so he's either plus over minus or he's minus over minus. We have information that tells us which one is not the case. Right. How would he have unaffected kids if he doesn't have the ability to pass a wild type allele on to them? So these plus kids down here had to get a plus from their mom and a plus from their dad. So we know he must be a heterozygote. If this is autosomal dominant. And so far everything is consistent. So we've been able to come up with genotypes that match the phenotypes that we see in the pedigree. So now at this point, the final question before you figure out whether or not this could be autosomal dominant is those 3 affected kids. Given the genotypes of the parents that we now know for sure, dad's a heterozygote, mom's homozygous wild type. Using those alleles from the parents, does it make sense that 3 kids, 2 females and a male, have this disorder? So if this is autosomal, there shouldn't be a sex bias. It should go equally to males and females, and that's basically what we see. About half the daughters are affected, about half the sons are affected. And overall, about half of the kids are affected. So that's consistent with being an autosomal dominant trait being passed from a heterozygous dad. So it could be autosomal dominant.
So if it's autosomal dominant, like, is the dad, would he just be like XY and then?
Right. So we don't. Yeah. So
we don't even look at the X.
Exactly. Yeah. You don't even have to worry about the X
It's
or
just
the
the
Y
gene,
gene. That you know he has an X and a Y
Yeah.
because he's a male, but the mutation is not on those chromosomes, so you don't have to write that into the genotype.
So it's just a gene that corresponds to that phenotype.
Right. So you could write in the sex chromosome genotypes,
Uh-huh.
but in this pattern, they're unimportant.
That's what I was getting really confused.
Yeah. Yeah, I know, and sometimes I do this too, and I confuse myself. I forget that, oh yeah, I was thinking it was autosomal, and I start writing down sex chromosomes, and then I get all screwed up. So yeah. Don't write down sex chromosomes unless you absolutely have to. It can cause confusion. Let's see. Okay, so same inheritance pattern. Could it be a recessive inheritance? It's not skipping a generation, which is why we didn't start with recessive as the first hypothesis. And so far we've said it could be dominant, but we just need to know for completeness. Maybe we can, maybe we can't rule out that this is recessive. So again, we just have to start with trying to assign genotypes. So if this is a recessive trait, could this be X-linked recessive? I can rule it out immediately, but I'm not saying that you should be able to jump to this conclusion automatically. There's something going on here that means it's not X-linked and recessive.
There's an affected man or 2 affected males?
It's the both. It's that there's an affected male and an affected son. So if this was X recessive, which I'll do in red, then the dad's genotype would have to be Y over X minus. He's got the X mutation on the X chromosome. Or does it make sense? Let's see, maybe it doesn't have to do with the male-male transmission. Or does it? Let's see. So this male's going to get the Y from his dad. We'll have to wait and see, I think. What does he pass to? So he passes his Y chromosome to all of his sons. He would pass the X chromosome to all of his daughters. So far, I think everything's okay. The sons have Y chromosomes, the daughters have X chromosomes. And if it's recessive, we can't predict the phenotypes there in the second generation based on just one allele. We have to know what came from mom. We do know mom's genotype. What's mom's genotype? Or do we? What are mom's possible genotypes?
Could it be the homozygous minus
Okay.
or heterozygous?
Right.
Oh wait, sorry. Yeah. That's it.
Homozygous, wild type, or heterozygous. If it's recessive, 1 minus or 0 minus means you don't have the trait. So she's either wild type or she's a carrier. Can the phenotypes of the second generation help us figure out her genotype? How did the affected son show up? This guy over here on the end. He's got, what's his X chromosome allele?
Minus.
It's a minus. So that had to come from a mom. So mom has to be a carrier. And since she's a carrier, she also has a plus X chromosome that she can give to her unaffected son. Unaffected daughter got which chromosome from mom?
Be the plus.
Right.
And she'd be a carrier.
And she's a carrier. And for the affected daughters, this is recessive. So if they're affected, that means they also got the minus from their mom. So this is where you see recessive. So this is totally consistent with a recessive inheritance pattern. And when you see the possibility of recessive showing up in every generation, it's because one parent's a carrier usually, or both parents. If both parents have the trait, of course you can show up in every generation, even if it's recessive.
So if it's X-linked recessive on like the dad that's affected, it's because that's the only X chromosome he has, so it's gonna show.
Right.
Okay.
Yep.
And with the unaffected daughter, the wild type is dominant.
Correct. Yeah, exactly. So when you have that X plus, that masks the effect of having a minus allele. But in a male, the Y chromosome doesn't have the X plus. He's got nothing. So even only one copy of a recessive allele shows up in the phenotype. You're right. Yep. So at this point, basically, we haven't ruled out any inheritance pattern.
If you don't see any skipping generations, do you automatically assume it's not recessive?
Sorry, there was a lot of negatives in there. If you don't see...
Oh.
Let's see. Say that again?
Okay, if you don't see a skipping generation, do you automatically exclude that it's a recessive?
So, you want to take that one? We automatically assume if you see it in every generation, the first hypothesis should be it's dominant. So you test that hypothesis, and if it turns out that that's not feasible, given the phenotypes, then the second assumption would be, okay, it must be recessive, we've got to figure out what's going on. Okay. But as in this case, we don't know if it's dominant or recessive, and it's in both cases it's totally consistent, either transmission pattern is consistent with the pedigree.
Okay.
All right? Thank you. Right. So this is a particularly unuseful pedigree for me to ask a question about because it requires you to go through this process for all 4 of the inheritance patterns and try to figure out, does it work or does it not? What haven't we tried yet? We did autosomal. We talked X dominant, right? We decided it couldn't be X-linked dominant because the dad would have to pass to all of his daughters. So it could be X recessive. Earlier today we figured out it can't be X dominant. Let's see. We started out with the assumption it was autosomal dominant, right? And that was consistent, if I remember correctly. So it's consistent with autosomal dominant, X-linked recessive. And if it could be X-linked recessive, then this is just something that I happen to know. We could work through it if you want. If it can be X-linked recessive, it could also be autosomal recessive. So basically, in this pedigree, we don't have enough information to figure out what the inheritance pattern is. It's definitely not X-dominant, but it could be one of the other 3. Which I know is intellectually unsatisfying, but hey, that's how pedigree analysis actually works in the real world. You get humans that have small families, and that's why, incidentally, a lot of the strongest pedigree analyses occur in families like Mormon families, where they have extensive pedigrees because of the history of the Mormon Church, and some other groups of humans like that, where there are extensive pedigrees and large family sizes, tend to be really strong candidates for doing phenotype and genotype mapping pedigree analysis.
So you said because it could be X recessive, it could also be autosomal recessive.
Right. So and there might be some rare cases where that's not true, but in general, if it could be X recessive, then you could just as easily explain it as an autosomal recessive trait. There might be some rare exceptions to that. I'd have to think about it. So the key, let's see, I think I can add that in here actually. Oh, but that's not the key. So here's the transcription and translation exercise. So what sort of...
My questions were I'm kind of confused on, like, how do you know, like, where it's supposed to start?
Okay. So let's start with that.
Okay.
So where does transcription start? And that relates sort of to the question about numbering the nucleotides.
Okay.
Right, question 3.
Yeah, and I also have a question.
So we'll go through as much as you want.
Okay.
So which nucleotide is the +1 nucleotide? It's the first one that gets transcribed.
Okay.
So the question really is, how do you know which nucleotide gets transcribed first?
Right.
So fortunately, we have the mRNA sequence, the transcript sequence. And so by definition, the first nucleotide that's found in the transcript, the RNA molecule, is the first nucleotide that got produced from RNA polymerase as it transcribed the DNA.
Okay.
So that makes that the +1 nucleotide. So really all you have to do is you take the sequence of the transcript and you line it up to the sequence of the DNA. and that's how you know the 5' most nucleotide in the transcript is the start,
Okay.
the transcription start site. Transcription start site. And then the nucleotide just to its left, the other one with the number sign here, is by definition -1. So we go minus to the left towards the 5' direction. And we go count up from +1 to the right. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. That's nucleotide 12. And there is no nucleotide 0 because you can't have no nucleotide. I mean, you don't go +1, 0, -1, in other words. It's +1 where transcription starts, and everything to the left is -1, -2, -3.
Okay, everything to the left of that is C at the top? Because there's nothing to the left of that.
So the numbering is all, that's a great question. The numbering is all in reference to the DNA.
Okay.
So we're numbering the DNA, as you see here on the left side, it says DNA coding and template right here. So the numbering only applies to the actual chromosome,
Okay.
the DNA sequence of the chromosome. The numbering makes no sense when you're talking about transcripts.
Okay.
Yeah, so it's all just, this is how geneticists define where on a chromosome the gene and the promoter is. So if we know that this C right there on the DNA sequence is the +1 nucleotide, we know we should look about some number of nucleotides upstream to find TATA, the TATA box. And we know that that's usually a certain distance away from where RNA polymerase starts transcribing, +1, the transcription start site. And I won't waste your time counting it, but it's, I don't know, estimate. That's something like 30 nucleotides. It's about -30. Well, it's the worst 3 ever, but that was the point of that.
Okay.
So you look that number of nucleotides in the minus direction, you should see somewhere around there TATA.
Okay.
If you're talking about a eukaryote, which we are in this case. It was set up in the question. It's a eukaryotic transcript, and so that's where the TATA box is. That TATA. We've got the +1 nucleotide, we've got the transcription start site. So let's see. So far we've got.
I'm sorry, where did you get the number 12 from?
So that's just 12 nucleotides starting from +1. So that's the first nucleotide, 2, 3, 4, 5, 6, I think, 7, 8, 9, 10, 11, 12. So yeah, that T or that A, depending on which strand of the DNA you're looking at, is the 12th nucleotide.
Okay.
And then you could do the same thing and figure out which number that is. Right, so that's 12. So that's the 24th nucleotide. Okay, so we've numbered the nucleotides. We've found the TATA box. I've added the 7-methylguanine cap, which wasn't on there in the first place, so that was kind of a mean thing for me to ask you to identify when it wasn't actually written on there. But remember, that was right there. It's added on to the 5' end of the messenger RNA molecule. Okay, so what else?
How come on the when the worksheet that you posted of this, you combined the untranslated region and the exons, and that threw me off. Like you just put plus this.
Okay, maybe we should work through this then, just a second.
So the pre-RNA, that includes everything except for the poly A tail that's in the mRNA and the intron?
So the pre-RNA contains the intron. So that's before any processing.
Mm-hmm.
So it won't have a cap, it won't have the tail, and it will have the intron.
But it won't have, yeah, so like for this example, then it'll go all the way until...
Right,
like,
you don't know, on the DNA sequence, you don't know how long it was. Yeah.
Okay, but then but the pre-RNA would just keep on going until like the DNA would
So
go?
it'll be about as long, right, exactly. Yeah, you don't know where transcription stopped initially.
Okay.
Yeah. So that's sort of it's a vague answer. There's not one right answer for that question.
And could you explain like what is like the poly A signal?
Yep.
What's the point of it? Because I always get confused with that because that's a termination signal, but then we still stop at
not
codon.
terminate. So don't confuse. Right, poly A tail does one thing, that's to transcription, and then we've got stop codons, which is translation. So just to make sure that there's a difference there. So we found the stop codon there by reading groups of 3, starting from the start codon until we reach the first stop codon. But then downstream of that, there will be the poly A signal sequence, AAU, AAA.
And does that always come after the stop signal?
Right.
Okay.
Should. Yep. And that's what defines, so what are the nucleotides between the stop codon and the end of the transcript? Right. That's the 3' UTR. And that's where you'll always find the AAU, AAA.
Okay.
So what happens is, during transcription, when RNA polymerase reads the DNA here, AAT, AAA, and it spits out in the transcript it's producing AAU, AAA. There's another protein that's hanging onto RNA polymerase that's basically reading the transcript as RNA polymerase makes it. And when that protein sees AAU, AAA, then RNA polymerase keeps transcribing. So it transcribes that much, approximately, more, about 20 nucleotides. And then there's an enzyme as part of this complex, another protein, that cuts the RNA molecule about 20 nucleotides downstream of the AAU, AAA. And that's where you see transcription stop. That is, there's no more correlation between the RNA and the DNA sequence. It ends right there where the poly A tail starts. So that's where you know, okay, this is absolutely where RNA polymerase basically stopped transcribing. And then after that, that poly A tail is added by yet another enzyme. It just adds on. That's all it knows how to do. It takes a 3' end of a transcript of RNA molecule and just starts sticking A's onto the end. That's all that enzyme does, poly A polymerase. That's its job. It grabs onto 3' tails of RNAs and just sticks a bunch of A's on them. So all of that is part of the 3' UTR. Again, because transcription stops at the stop, translation stops at the translation stop codon. And that AAU, AAA is related to transcription, the production of the messenger RNA in the first place, and it's always past the stop codon. Hence, it's always in the 3' UTR.
And then the splice sites and the branch point, that's just talking about the intron again?
Right. So we've got, so we found the 5' trouble talking now. We found the 5' and 3' splice sites. Right, those were there in the intron, and the branch point, some sort of an A. found the branch point. We found the poly A signal sequence. That was AAU, AAA in the RNA, or AAT, AAA in the DNA. We found the poly A tail. We found one intron. We found exon 1. That was everything from the start of the transcript to the first intron. So where's the second exon? What are its boundaries?
Up until the stop codon, like.
Well, like the exon 1 includes the 5' UTR. Exon 2 includes the 3' in this example.
Oh, okay.
Right, so the exon does not just mean regions of a transcript that encode protein.
Oh, okay.
It's basically anything that's left after intron. After splicing, everything that's left we define as having been part of an exon. So there are 2 exons in this case, separated by a single intron.
So exons include the
The UTRs, yeah.
Okay.
Yep. We found the promoter, we found the UTRs. We talked about, although we haven't written the pre-RNA sequence, but that's everything starting from the transcription start site and including the intron sequence.
Mm-hmm.
And then not including the poly A tail. So in other words, it would be the sequence of the DNA coding strand from the +1 nucleotide all the way through the intron up until where you see the poly A tail start. That would be the sequence of the primary transcript. There would be U's instead of T's because it's RNA.
Could you do number 2?
Yes, let's do number 2. That's probably the next most important. The 5 through 8 are sort of thought questions. We could talk about them, certainly.
I get, like, what codes for proteins, but it was just, like, which element is transcribed.
Yeah. So basically, we've already actually figured out which elements are transcribed.
Does that mean, like, what shows up on the mRNA? Or the pre-RNA?
Right. So everything that's part of the pre-RNA, that's the answer to question 4, was something that was transcribed.
Oh, okay.
Yep. So the answer to question 4 basically is the answer to the first part of question 2, what's transcribed. So again, that's everything from the +1 nucleotide all the way through any introns and up until you see the poly A tail start. So that's the pre-mRNA sequence, and that is everything that's transcribed.
Okay, yeah, that makes sense. That's why the splice sites are transcribed, because they're in the pre-RNA.
Right. Yeah.
Okay.
So that's the main point of my asking that question, is that sometimes students get the idea that introns aren't part of the messenger RNA molecule, or the RNA molecule originally. So I'm just trying to reinforce that, yes, RNA polymerase does go through the process of making a transcript that includes all of these introns, and then the cell just goes ahead and cuts them all out.
So if we're talking about all the elements, then so the 5' and the 3' UTRs, that's transcribed.
Correct.
The introns are transcribed.
Yep.
The exons are. The
Yes.
Tata box and the promoter aren't.
By definition, right, because they're upstream of the transcription start site.
Yeah.
They're to the left of the TSS, or the +1 nucleotide.
And then the splice sites are, and the branch point are transcribed.
Yep.
The poly A signal is, but the poly A tail is not.
Correct.
And then the 7 mg cap, that's not.
That's not, because it's not a nucleotide. And
Yeah.
It is kind of, but
But it shows up after.
details. Yes, it gets added after transcription.
Okay. And then what codes for a protein? The UTRs don't, right?
Correct,
And
yeah.
the introns don't, but the exons do. The Tata box and the promoter don't.
Well, the exons, parts of the exons code for protein.
Oh, because UTRs are part of the exon.
Right.
Okay. And then the splice sites, they don't.
Right, because they're in introns, and they get taken out before translation happens.
Yeah, and the branch points don't.
Right?
The poly A signal and the tail they don't either, right?
Because they're in an untranslated region.
Okay.
Yep. Whew. Yep.
Then what do you mean by question 7?
Do you understand the idea of starting reading a messenger RNA molecule at the 5' end and looking for the first AUG left to right, like a ribosome would, and then you start translating in groups of 3 nucleotides?
Oh, okay.
So yeah, it's a poorly written question, or a goal rather. How the messenger RNA is translated by ribosomes into a sequence of amino acids. In other words, can you take those codons that I underlined there in red, AUG, GCC, CCC, and use a codon table to tell me what the sequence of the resulting protein would be? I.e., question 8, or goal 8, can you actually translate this messenger RNA?